Glycolysis is a breakdown of glucose which produces pyruvic acid and releases energy. Glycolysis will take place in the cytoplasm of both eukaryotic and prokaryotic cells. In order for glycolysis to begin, it will need ATP. The starting reactants will include ATP, glucose, ADP phosphate, and NAD+. Ending products will then turn into 4-ATP, NADH, and pyruvic acid. Glycolysis uses a substrate-level phosphorylation. This means that the substrate-level phosphorylation is a metabolic process in which ATP is produced through another ATP. Further, it will use NAD+ as an electron carrier, and it will also be categorized as a carbon rearrangement since it does start off with 2 ATP and ends with 4.
This is step 1.5. Pyruvate is a chemical compound in biochemistry. Pyruvate is the main output in glycolysis and the main input for citric acid cycle. From the cytoplasm, pyruvate is transported to the mitochondrion where further oxidation occurs. Before the citric acid cycle can begin, pyruvate must be converted to acetyl-CoA. And for this to happen, NAD+ must be converted to NADH. Coenzyme A has to be into the reaction, and CO2 must be released from these reactions. Acetyl-CoA is a factor which links glycolysis to the citric acid cycle. This step is carried out by a multi-enzyme complex.
The citric acid cycle, also known as the Krebs cycle, is joined with CoA, which is a 4-carbon molecule structure, and it releases CoA and it forms a 6-carbon molecule called citrate. Step 2. Citrate is converted to isocitrate by first the removal of a water molecule and then the addition of a water molecule. The isocitrate is then oxidized, which releases a carbon dioxide molecule, and the NAD+ turns into NADH. And then the molecule that it turned to was a 5-carbon molecule, which is called A-ketoglutarate. The A-ketoglutarate is then oxidized, which releases a carbon dioxide molecule, and then it picks up a acetyl CoA molecule, and the NAD+ turns into NADH. The A-ketoglutarate molecule is then oxidized, which releases a carbon dioxide molecule and picks up a coenzyme A molecule, and it forms into succinyl CoA. Next, the CoA molecule from the CoA succinate is released, and in replacement, it has a phosphate group that attaches with it and is then transferred to the ADP to make ATP, and also the 4-carbon structure that's formed at the end is called succinate. Succinate is then oxidized, which forms fumarate, and then during this reaction, 2 hydrogen atoms transfer into FAD, forming FADH2. Water is then added to the fumarate, and this makes it turn into another 4-carbon structure called malate. Oxaloacetate is then regenerated by the oxidation of malate, and then the NAD+ is converted to NADH.
Electron transport chain, which is also known as oxidative phosphorylation, is a metabolic pathway in which electrons are transferred from electron donors to electron acceptors in redox reactions. There are 4 protein complexes. Complex 1 pumps 4 hydrogen ions from the matrix into the inner membrane space. Complex 2 receives fat H2 and delivers electrons to the electron transport chain. Complex 3 passes protons through the membrane and carries electrons to the cytochrome. Complex 4 reduces oxygen, then picks up 2 hydrogen ions to create water.